So today topic is synthetic biology, guided microbial consortia for enhanced biofuel yield. So in this topic, what we discuss like, actually the microbes, they are genetically modified for some specific purposes. Even microbes can be used as a biofactory. It can make some valuable compounds. So similarly, the microbes will be modified, but with the help of the synthetic biology approach. And the next part is, you can see here, it is in topic microbial consortia. Usually we modify the individual microorganism, and here we'll try to modify more than one microbes, different type of the microbes. Because if we take the consortium, then the load will be distributed. The specific microbes will perform the specific task. So the microbial consortia, that's why I've taken the consortium here. And third part is the biofuel. This is a demand, the renewable energy demand today. So every country is having the demand of the biofuel. And you already know the US and Brazil, they are the largest producer of the ethanol. And it is obtained from the sources, the living sources, the food crops, right? And they are trying to blend with the gasolines and other petroleum products. So actually what happened, we are trying to produce the butanol from the lactic acid bacteria, but I found the production yield was very low. And that's why I decided to do something different. The yield could be increased. So now the synthetic biology, this is the emerging field. And this is not only, we can say it is a core biology, we can say it is an interdisciplinary approach. And why I'm saying the interdisciplinary? Because the synthetic biology use the techniques and tools which is based on the engineering cycle, design, build, and test. So this we'll discuss into the later PPTs slides. So I just minute. Right. So I'm targeting here the compounds, the valuable compounds, the either ethanol or butanols, and which has to be produced from the microorganism. And that there are different generations, that there are different generation of the biofuel. But the fourth generation, we targeting the fourth one, the fourth generation. So fourth generation, particularly the modified organism. So here again, we modify the organism. We put the genetic circuit into the microbes, and we see what happens. Actually, I don't know, but we have a hope that we can get the better yield, right? If we see the global market of the biofuel, see the two countries, North America and Europe, you can see, they are the largest producer of the biofuel. And if you compare with the 2022 to 2029, we are expecting
पॉइंटर का ऑप्शन वहां कोने में होगा, उसको क्लिक कर लेंगे तो आ जाएगा. पीपीटी के जो लेफ्ट कॉर्नर में आप करेंगे ना तो वहां आएगा उसको. हाँ, वहां पर में एक बार राइट क्लिक करिए. मुझे लग रहा है वहां पॉइंटर आना चाहिए. हाँ हाँ हाँ, उसी में देखिए तो पॉइंटर है, दिख रहा है.
एक मिनट
जी, लेजर पॉइंटर या पॉइंटर कुछ होगा एक बार. हाँ हाँ हाँ, हाँ हाईलाइटर कर दीजिए. यस, हाईलाइटर चलेगा ना. यस, नहीं हुआ.
जस्ट मिनट. ओके. हाँ. Is it with just
है उसमें हाइलाइटर?
is it visible?
हाँ अब ठीक है, वो चलेगा.
So just we can see here actually in 2022 the market size was 135 US billion dollar, and we are expecting in 2029 the 5% increase in market, the biofuel market 189.96. So every year the market, the biofuel market is increasing and increasing, not only into the US and Europe countries. If we look at it into India also, you can see this the 2022 and 2023, 2023, the 12% ethanol that is used as a biofuel, that blending to the gasoline. So if we compare with the 23, 20, 14, 20, 13, 14, and 22, 23, you can see the increase. It was 2%, now 12%. The gasoline is blended with, the gasoline is blended with 12% ethanol, right? So it is demand, huge demand, and therefore we uh, targeting the production of biofuel with the help of the genetically modified, with the help of the engineered bacteria. So see, you can see the two diagrams. In the top diagrams, you can see this is just like monoculture that can make the biofuel, ethanol, butanol, whatever actually, the any desired products. And the bottom diagrams that says the mixed culture, the many microbes, different microbes, they are working synergistically and that make the product. So in the top one, just right. So in the top one, you can see the yield is very less, and in the bottom one, yield is very high. And what is the reason of having the high yield if the mixed cultures is used for the production of the desired compound? The reason is in the monoculture, there would be a possibility that the overburden, or we can say the metabolic burden, some of the toxic compound can be formed. Maybe the the limitation of the resources can decrease the yield. It is not producing the up to mark the desired yield. But if it is mixed cultures means you are distributing the load. The same thing, if you distribute the load into the different individual, and each individuals perform their specific task, loads are distributed, so the performance would be better. And the same thing is happening in the case of the mixed culture. The mixed culture loads are distributed. Different microbes are used in one pot, one tube. We have taken one tube and we taken the different microbes. And the initial substrate that is fermented, or that is what we call processed by the microbes one, that make the product, and that products will be the initial substrate. This product will be the substrate for the microbes 2, and microbes 2 will make the again product, and this product will be substrate for microbe 3, and finally you get the better yield of the, the specific or desired compound. So this is the strategy, right, that we can apply in future research. But the thing is, again, if you take the mixed culture, so a lot of the things will be happen. There will be a competitions between the microbes. So what we can do, therefore the the microbes will not will communicate to each other symbiotically, synergistically. So synthetic biology approach will use to for this. Therefore, they can communicate synergistically. So even in the drastic condition, the environment is highly variable. The mixed culture will be able to produce the good quantity of the yield, the product, desired compound. So, these are the challenges you can see with the monocultures. The metabolic burden, chemical production, and intermediate accumulations will often cause cell stress due to chemical toxicity. So the accumulation of the compounds will cause the cell toxicity. And third one is, you can see the consumptions and additional ATP and NADPH leading resources shortage. So many biochemical processes or metabolic pathway require the energy power, ATP, reducing power, NADH. But if you have a cascade of the metabolic pathway, multiple pathways, 1, 2, 3, then there is a chances of the shortage of the ATP and NADH. And this is possible in the case of the monoculture. But if it is mixed culture, then somehow we can control it. So what is the solution? The allocation of limited resources for different tasks. Again, this is also problem. Limited resources. So solution is again the mixed culture, a microbial consortium. So you can see in the right side, the polysaccharides from biomass, this will be processed by the cells, the microbial consortium, but this microbial consortium is the programmed cells, not one, multiple programmed cell. And then this will be processed, and then they will give the output, and output is the target chemicals. So this is the strategy. We have to construct or develop synthetic microbial consortium that can convert the biomass into the target chemical like ethanol, butanol, that could be used as a biofuel. Right. So synthetic design, that is not very simple. You can see here. You know, suppose we design a car, and for designing a car, you require different parts, and you assemble the different parts together, and finally you'll be able to design a car. But for designing the car, you have to, no, you can build the car. So for first you have to have a design of the car. You have to have a design of the car, and then you build the car, and you check the car, performance of the car, right? Design, build, and test. Same thing in the synthetic biology, we do it. We design, we build, and we test, right? And then we learn. Learn means you have a design, and you build this like you have a design of the pathway, metabolic pathway. You build that metabolic pathway, and you check that metabolic pathway into the chassis like E. coli or Saccharomyces cerevisiae. And if you do not get the the desired proper, or you do not get the proper performance of this chassis, then what you do, you can redesign it. You can redesign it. Oh, this is the problems that we did it in my design. You have done some mistake in the design. You change the some design somehow again try to build the pathway and put this pathway into the microbes and see the performance of the microbes. If microbes is doing the better performance, then we can say your design is the correct design. So synthetic biology use the same, this is the engineering principle, design, build and test, design, build and test, okay? This will be repeated and repeated and repeated. And once you achieve the proper performance, then you can stop this one, this cycle. So if I say synthetic biology, there are different parts. The different parts means like the promoter is a one bio parts. Operator is the another bio parts. Gene is the bio parts. Different genes are the bio parts. Okay? So ribosome binding sites is the bio part. And these are the bio parts that we can assemble together. Once we assemble together, then this can make the bio device. And some of the other bio parts like polymerase, and this can interact with the bio device. And then we see the performance of the device. This complete thing we call one module, right? So synthetic biology here, we are not using the genetic engineering approach. You know, once the synthesis cost was decreased, DNA synthesis cost, so the people are more mainly focused onto the synthesis, not like the doing the genetic engineering things. So because in the genetic engineering things, there is many chances of the failure, right? So maybe like you have to digest it, cut it, ligate it, transfer it. So each and every steps you have to face some of the failure. So it's easy to, we can get the complete things synthesized, the complete cassette, genetic cassette synthesized and put into the microbes that we call chassis factory, microbial factory. And we see the desired, this microbes making the desired product or not, right? So that's why we call this is the synthetic biology. The synthetic biology aims to redesign and rebuild metabolic pathway in order to synthesize a specific molecule from cell factory. And here you can see in the bottom, these are the different parts, they are assembled together, right? What you are seeing in this one, this diagram, you can see here, they are the substrate, initial substrate, which is into the microbes. Suppose we take the example of E. coli. So suppose these are the initial substrate, all green color one. They are already present into the cell. And after this, you can see this can make the different product. So multiple biochemical reaction, metabolic pathway, right? And after that, we suppose this is the desired target one, the target last one is the target product. But this can be formed from the multiple different pathway. We have to select only one or two pathway. That could be simple, not like this can make one product and this product can interact with the other metabolic pathway. This can interact with the third one, okay? So there will be like, if this is the case, then will not get the achievable yield. So we have to select the pathway which is minimal pathway, right? there is a minimum crosstalk with the different pathways, right? So here we can select, like you can see this one blue color one. So there is no such much interaction with the other pathway, the intermediates with the other pathway. So we can select some minimal pathway from this one, right? So this is also important, like the selecting and ranking the pathway is important. Which pathway we should select it, which has a higher ranking in the pathway. There are different softwares that we can use it for selecting the pathway. And then once we have this pathway, then we can build this pathway and transfer into the microbes to get the product, right? So one examples you can see here, the final product is 3-hydroxy propionic acid. 3-hydroxy propionic acid. And there are two organisms which is used here, microbes. One is Vibrio and another one is E. coli. We have engineered the E. coli. Vibrio is the natural one, wild type, but we engineered the E. coli for the production of the hydroxypropionic acid. Alginate is the initial substrate for the Vibrio species. Alginate is the initial substrate for the Vibrio species. This makes the product acetate, and acetate is used by the engineered E. coli cell and make the hydroxypropionic acid. So gene which make the enzyme, and these enzymes, the gene of that enzymes are, the different genes are transferred into the E. coli cell.
हाइलाइटर आपका नहीं काम कर रहा है?
नहीं कर रहा है काम?
नहीं, हाइलाइटर से तो नहीं दिख रहा मुझे.
Okay.
एक बार देख.
So, you now abhi?
हाँ हाँ हाँ
Right.
हाँ.
So you can
No.
see this is the some of the genes, some of the enzymes which is not in the E. coli for the production of the hydroxypropionic acid. So that is transferred into the E. coli with the help of the plasmid, right? And that make the 3-hydroxypropionic acid. But the problem again here, what is? Actually, it is not synthetically designed. It is engineered E. coli. It is not synthetically designed. So what is the problem with this cells? Can you see into the E. coli? This is the E. coli cells. This is the Vibrio cells, right? Everything is in one cells. There is no two cells. There are different cells, the E. coli and Vibrio. This is the consortium here. So what is happening here? They are having competitions with the same substrate. Can you see the competition? The Vibrio and E. coli. But the Vibrio is this one, if you see, this is more compatible for this substrate, right? So therefore, the lack of this one, the scarcity of this substrate, the E. coli population is decreased. E. coli, green one, E. coli population is decreased. And once the E. coli population is decreased, because the E. coli that make the 3-hydroxypropionic acid, the product, the red color one, this yield is decreased. Product, this yield is decreased because of the competitions, right? So how to solve this? So we can design a synthetic circuit, you can see, and put into the E. coli. This is the synthetic circuit. And put into the E. coli. And what is this circuit? This we call the population guider. The population guider. And this population guider, what happens? Like if in In medium, ampicillin is present, and in the medium, the E. coli and Vibrio, both bacteria are present. This ampicillin kill the Vibrio bacteria. It will kill the Vibrio bacteria, right? So it kill the Vibrio bacteria, but E. coli, if E. coli make the enzymes which can degrade the ampicillin, this E. coli, green color, this make the enzymes which can degrade the ampicillin. So once ampicillin is degraded, then this bacteria will survive. The Vibrio bacteria will survive. So Vibrio bacteria is benefited by the growth of the E. coli, because once E. coli grow, it make the one enzyme that can degrade the ampicillin, and once ampicillin is degraded, then the Vibrio bacteria will survive. So we design a synthetic cassette in a way that can make the ampicillin degrading enzymes, right? So how you can see this the red one, this is the 3-hydroxypropionic acid. Once the E. coli make the 3-hydroxypropionic acid, and one of the regulator, that is the HP sensitive. So this regulator interact with the 3-hydroxypropionic acid and attached to the promoter. This promoter will express the ampicillin degrading enzyme, this BLA, right? So this will be expressed, and this is expressed once the regulator plus 3 HP that can interact with the promoter. The block which is under control with this promoter, 3 HP sensor promoter. So once they interact with this promoter, then this will be expressed. So even the bacteria is in low population, E. coli bacteria, but this will express the ampicillin degrading enzyme, due to which the Vibrio bacteria will survive. And Vibrio always will try to keep the population, the population of E. coli high. Therefore, the Vibrio bacteria can survive. So there would be some cooperation between these two bacteria. Okay. Otherwise, if the E. coli population is not high, the Vibrio bacteria will die. So there should be cooperation. So Vibrio bacteria is looking its benefit, and therefore this is the E. coli is also surviving it. So this is possible because of the new synthetic designs which is transferred into the E. coli. So you can modify yourself. What actually depends on you how to make the synthetic circuits and transfer into the bacteria for specific some purposes. So that's why there's the competition, and these problems can be resolved if you transfer such type of the circuit into the bacteria. Next, you can see, the biofuels can be produced from the food crops also. But we are targeting again here the lignocellulosic biomass. And the reason again here why we are getting lignocellulosic biomass, first thing is, after hydrolysis of the cellulose, we get the both sugar, carbohydrate, pentose and hexose. So we have pentose and hexose sugar both. The microbes can utilize both different type of the sugar and can make the good yield of the compounds, ethanol or butanol. Yeah. So this is the good thing. But the bad thing with this lignocellulosic biomass is the cellulose is what we call, it will not degrade it easily. It is even in the high temperature, it could not be degraded easily, right? So what we can do again, because it should be degraded easily, And after degradation, the sugar which is produced, the carbohydrate which is produced, could be the produce should be utilized by the fermenter, for the microbes which is involved in the fermentation, right? So one of the, like, if suppose we have a design something like that, one microbes can degrade the cellulose, and the another microbes can use the degraded product like glucose, xylose, arabinose, and that can be fermented by the another microbes, and these microbes will make the butanol or ethanol. So the design which we have, you can see in the diagrams, like you can see the module 1 and 2, module 2. The module 1, that is cellulase producer. The cellulase is the enzymes which can degrade the cellulose. And you can see inside this microbe, this is the genetic circuits, right? And this make the cellulase. And this cellulase will degrade the cellulose. And then this glucose, once it is degraded into glucose and some other carbohydrate, this glucose is utilized by the module 2, and this we call chemical producer. This will make the butanol, chemical. This will make the butanol. So after fermentation of the glucose, the butanol, ethanol, or some other product can be formed here, right? Is it possible like we can design it such design? So we can design. The problem again, you can see the individual better the microbes which is engineered for, like in one microbes we try to put the synthetic circuits of cellulose producer and chemical producer both, but again yield was less. Therefore, uh, some researchers, they try to make the consortium, like use the two synthetic microbes together for the better yield. So this is the one thing that we can do. The another thing you can see here, right? What is it? This one more thing you just we can think, like
Please
suppose,
close your camera, Dhirendra Dahiya ji and Sinha ji. Please continue, sir.
yeah. Now, see, like one of the microbes, like we used two microbes. One microbes produce cellulase enzyme, and another microbes is the chemical producer that make the target biochemicals, target product. Is it possible, like the microbes which make which degrade the cellulose into the glucose, and there is a chances that glucose can also be utilized by the cellulase producer enzyme, like this microbes, the module 1 cellulase producer. This microbes itself can use the glucose and ferment it, right? But we don't want that the glucose should be used by the module 1 microbes, the cellulase producer microbes. Because if it is used by the cellulase producer microbes, then the for module 2 chemical producer microbes will feel little less glucose for fermentation. Okay. And we want the high amount of the glucose should be used by the chemical producer, not cellulase producer. So how we can resolve this problem? So again, you can see this is another synthetic design which can resolve these problems too. I'll tell you what is it. So, so what we can do here, you know, uh, just me. This blue color you can see, this is actually we call auto-inducer. Auto-inducer actually that is involved in the column sensing signaling pathway. This auto-inducer is a AHL, acyl what we call hydrolactone, a homolactone, like acyl homolactone, right? And this is a cellulose. This is a cellulose. It is not degraded yet. This is a cellulose. And if it is a cellulose, this is not used by the this is not used by the microbe one, because it is cellulose. It is not changed into the monosaccharides like glucose. So it is not utilized by the microbe one. And the microbe 2 also not utilize this one because it is not changed into the monosaccharide. In the microbe 2, you can see this is the synthetic circuits which make the chemical, which make the biochemical, which make the desired product. And the microbe 1, I'll tell you in the microbe 1 also there would be some synthetic circuit. Okay. So this is the AHL which is produced here. This AHL you can see auto-induce your cell, auto-induce the bacteria itself. It is induced itself. So in this case, what is happening? The AHL is amplifying it here. The more AHL is produced, and this acts as an auto-inducer. It induces the cell itself. And during this process, inside this bacteria, you can see enzymes are produced. These enzymes can degrade the cellulose. This can degrade the solid cellulose. You can see this can degrade the cellulose, these enzymes. Right? So now this is just the production is start here. The production is start. Still it is not degraded, but the production is start here. And but this AHL that induce the genetic circuit into the second microbes. So the genetic circuit is switch on into the second microbes. In the first case, second microbe it was switch off, but in the second case, this is switch on because of this inducer AHL. This is switch on. Circuit is switch on for production of the biochemical, but the microbes is not able to take the initial substrate like glucose. Okay. So now you see due to the AHL, here high HL, there would be some genes that can degrade the cells, the first microbe cell lysis, we can say. This will be more clear into the next slide. So some of the genes that make some enzymes that will degrade the cells. The first one, the cellulase enzyme producer cells, is degraded here, and then these microbes, these enzymes are released, and once the enzyme is released, then this can hydrolyze the cellulose, and once it is hydrolyzed the cellulose, then it is accessible for second microbes, and then second microbes, because it is switch on condition, so second microbes get the substrate and let it make the product, desired product this way. So this case, what is happening? The first is cell is lysed. So cell will not utilize, the first cell will not utilize the glucose molecules. Cell will not utilize the glucose. This glucose is not utilized by the first cell. It is broke. Cell is lysed. Cell is dead now. But the second cell, the biochemical cell, biochemical producer cell, will utilize the glucose. Right? So this will utilize the glucose. So all the glucose, all the monosaccharides which will be generated after hydrolyzing will be utilized by the second microbes. Right? So now you can see this is more clear here in this diagrams. So in these diagrams, this is the same thing actually we explained here. Only thing is we have put the genetic circuit into microbes. One microbes will make the enzymes, the another microbes will produce the target chemicals. So you can see here, if you try to understand this genetic circuit, can you see this is the one genetic circuit, and the top one in the first microbes, one genetic circuit, and in this genetic circuit, the LuxI protein, this LuxI gene is under control with the promoter P-LuxLec-O. So this promoter make the LuxI, but how it make the LuxI? Once this is the LuxR repressor, the LuxR repressor can repress the expression of the LuxI, but LuxR repressor, it is bound with this, the blue color one, this is AHL, this is AHL. So once this Lux repressor, this one bound with this AHL, then this can activate the expression, bind with the promoter and activate the expression of LuxI. LuxI formations, once the LuxI is formed, this LuxI and S-adenyl methionine plus acyl ACP, this will make again AHL. This will make again AHL, auto-inducer. So now the LuxI, this is responsible for making the AHL, auto-inducer. The auto-inducer will be formed here, right? Synthesized here. The another thing you can see in the same cassette, there is the choline and endolysine gene. Choline and endolysine gene, this is specific for the cell lysis. This protein is specific for the cell lysis. So once these genes are expressed, this can degrade the cell. But again, you can see the another circuits, these circuits that make the BGLC protein, BGLC enzyme. BGLC is a beta glucosidase enzyme. And this beta glucosidase enzymes will hydrolyze the cellobiose. This is a cellobiose. Okay. So this hydrolyze the cellobiose. So this produce the beta galactosidase enzyme, and that will be released after cell lysis. So these two things that are under control, they are in the control. So this is the top one, first circuit, that is involved in the cell lysis, and the bottom these circuits involved in the production of beta glucosidase enzymes. And once the beta glucosidase enzyme is produced in the proper amount, cell is lysed, and this beta glucosidase enzyme dissociate the cell bios into the monosaccharides, and this will be utilized by the second cell, the biochemical producer cell. You can also see here, it is like that one thing is this one, another thing is the glucose which is produced. This event cannot be utilized by the cell if the sum of the cells, if it is not lysed, then it is not utilized, because the transporter proteins which help in transport of the glucose into the cells, this is mutated. Gene for transporter protein is mutated. So the glucose transporter, the mannose transporter, galactose transporter genes, like it is mutated. So finally, the glucose will not be utilized by the first cell. So all in all, glucose molecule that is present for second cells only. So now the second cell is only responsible for the biochemical production. Then second cell will make the biochemical, the desired compound. So, and GFP we put just like you can even quantitate it. What is how much quantity of the product is formed. So this is the synthetic design. So now what actually I want to try to tell you here, we can also think it about this one, like the butanol producing pathway. So we can also design the synthetic circuits for the butanol productions, and we can increase the yield. So two things we have discussed here. One is we are using the lignocellulosic mass, biomass, and this will be degraded by the one of the factory, one of the cells, microbes, and another thing is the another microbes that will be used for the production of the desired product. So this is the application of the synthetic biology. Now, now you see, uh, sometimes what happens, like if you want to engineer one of the cells, like lactic acid bacteria, or you want to engineer the E. coli cell, or you want to engineer like Saccharomyces cerevisiae for the production of the desired compound. That cell is only taking the glucose as an initial substrate for production of the ethanol or butanol, and it is not utilized. The pentose or if the cells is able to utilize all the type of the sugar, then that cell can perform better way. That can make the better yield, produce the better yield. So this is also like these circuits explain you like how we can modify the cells that can utilize the different type of the substrate for production of the desired product. Right. Now we come to this one, this part. Right. So actually this is my work, research work, but this is the pathway that is given here only. Uh, you can see the bottom one, this part. Actually, these are the enzymes which is not present in the lactic acid bacteria. Lactic acid bacteria make the ethanol, but it cannot make the butanol. And our objective was to make the butanol. So as compared to ethanol, this is high energy rich compound, butanol. So our objective was to make the butanol. So what we did, we have this is genetically actually we have genetically engineered, genetically modified the lactic acid bacteria, and we use the two plasmids. In one plasmid, we have cloned three genes. These three. Another plasmid, we clone the later the downstream genes and transfer into the bacteria, lactic acid bacteria. Still, we didn't get the good yield. Then we thought, why we are not getting the good yield? So the lactate and ethanol. So what we did, we have blocked the pathway. This pathway we blocked. We blocked this pathway. So we block this pathway. How? We modified, we mutate this region enzymes, which is responsible for lactate and ethanol production. And then we checked. Then we didn't get again good yield. The reason was the growth was now decreased, the bacterial growth, lactic acid bacteria. Then we thought that if this lactate is involved in somehow in the growth of the bacteria. So we didn't block the pathway completely. We downregulate the genes which can make the pyruvate, right? So we direct this metabolic flux more towards this direction, and less towards the lactate direction. The yield is increased, but we want to increase more, and therefore we thinking it like use can we take the help of the synthetic biology to design the new microbial consortium for the production of the butanol. If it is possible, then we may be able to achieve the desired yield. So this is all about the synthetic biology.